Let's talk about probiotic microorganisms. As we have been discussing about probiotics, and we need to understand that what are the different microorganisms that can be classified as probiotics. So, if we look at the history of development, they have been used from last 40 years, especially from Japan and more than 30 years in Germany, that they have started developing food-based products that contain the probiotics of human origin. By human origin, it means that either they come from human origin or either they are known to be there in the human GIT tract and having their own functional properties very much well known and understood in human beings. If we look at these viable probiotic strain that would have these functional properties, as we earlier said that this is what all the probiotics should have. They come from a wide variety wide variety from different genera, from different strains, and from different species. So it doesn't mean that all the probiotics have to come from one group of microorganisms. Rather, this probiotic potential is found in many species, in many genres, and in many strains, and it is variable in every strain or genre or every species. So we have to actually look for, isolate, study, and define what is the best suitable strain for our products that can be commercialized and that have got more potential health benefits as compared to one specific group of probiotics only. So if we look at the availability of these products, as we earlier said, they are mostly in food or nutrition or pharmaceutical areas. These are the different preparations, and mostly they apply lactic acid bacteria. So these are by mean that they can ferment the lactic acid and produce they can ferment lactose and produce lactic acid. That is why they are called as lactic acid bacteria. So this is one of the criteria to define whether a microorganism is a lab or lactic acid bacteria or not. But it doesn't have to be lactic acid bacteria all the time. For example, we can have some non-lactic strains as well that can have functional properties or health improving properties, and they can be classified as probiotics. The examples include Bacillus cereus, Escherichia coli, and Saccharomyces cerevisiae. So these are again used as probiotics. As you know that Bacillus cereus or some strains of E. coli are pathogenic as well. So we have to actually look for certain strains that have got within these species or genera that they have probiotic potential. So they are present in the market already. The one that I earlier discussed now, they are present in the market and they are present in the form of pharmaceutical preparations, and they are used for animal feed as supplements. So we know that they have got health benefits that they incur to animal when they are given this kind of animal feed. It might be improved health or improved yield of meat or other valuable commodities such as milk from animals. So this is where we know that probiotics have been already marketed in the market for animal feed. If we look at this process, is a lengthy process to make up a particular strain, to test and identify that this strain can be probiotic, and then selling that strain in the market. It needs a lot of regulatory processes, and it is a very lengthy research and development process, as well as regulatory requirements have to be met. So generally there are three major requirements that must be met by a producer of probiotics before they can sell any product into the market. They must first of all give enough scientific evidence about the efficacy of these probiotics, and this efficacy is not only the claim efficacy, but it must be substantiated with clinical trials as well. So if it has been clinically tested and proven that they have got some efficacy, these strains, then they can be one passing point for these strains to be used as commercial product. The second and very important aspect is safety for consumption. Whether these probiotics, whether they are safe or whether they are not safe, this is one of the very much deciding factor to approve any probiotic for the commercial use. So if they are not safe, of course, there cannot be any question about approving such probiotics for commercial applications. Then the third element is analytical determination of the effective components of these probiotics. So we should be able to substantiate the health benefits by certain diagnostic tests, and we should be able to see that this benefit is actually derived by production of certain components that can be related with consumption of these probiotics in clinical studies, in human trials, etc. The functional properties of food products, again, it comes from different bacteria. So normally the commercial products, they can have combination of different strains together, and where they can have synergistic effect, or where it might be possible that every strain would give a certain extent of benefit to the end user. There is another term used. This is called as biotherapeutics. This is basically the therapeutic agents coming from the biological sources, and they are normally based on certain strains, probiotic strains that have been tested, proven in the clinical studies. Generally, the ones that are available in the market, they are lactic acid bacteria. But there are other examples as well. As I said, that there's new research as well, and we've got increased capacity to understand the other kinds of probiotic bacteria. For example, E. coli, Saccharomyces cerevisiae, and Enterococcus faecium, or E. faecalis. This is the different other types of biotherapeutics available in the market having probiotic potential. So this was some information about different probiotic microorganisms, or microorganisms having probiotic potential.